Hello, everybody. My name is Benny Mosqueda. I'm in third year undergrad at the University of California, Santa Cruz, studying molecular, cellular, and developmental biology. At UCSC, I'm a part of the Carpenter Lab, and I'm super excited to be presenting at the Camp Symposium this year. My project is titled Determining the Expression Levels of LincRNA COX-2 in Resident and Recruited Alveolar Macrophages. Carpenter Lab studies the innate immune system. The immune system is divided in two halves. There's the adaptive immune system, primarily composed of B and T cells, and its main function is to produce antibodies. In the Carpenter Lab, however, we focus on the innate immune system, and more specifically, we look at macrophages. Macrophages are a type of white blood cell that engulfs and digests cellular debris through a process called phagocytosis. The name of macrophages actually implies the function, as macro means big and phage means eater, so they're literal big eaters. In our introductory level biology courses, we are taught the central dogma of biology. DNA is transcribed to RNA, and RNA is translated to protein. Now, while this is a very important biological process, it isn't the only, and actually only 1 to 3% of the mammalian genome has protein coding capabilities. Of RNAs, long non-coding RNAs are the biggest subgroup, and as the name implies, do not encode for protein. So initially deemed without having any function, many scientists, including me, are now trying to understand their role, and in our case, understand their role in the immune system. So the characteristics of lincRNAs: they must be longer than 200 nucleotides, they must have tissue-specific expression, and have no open reading frame, or ORF for short. For my project, I'm specifically looking at lincRNA COX-2. LincRNA COX-2 was first found by Susan Carpenter, the PI of my lab. Shout out to you. By doing a plus and minus PAM stimulation on bone marrow-derived macrophages, and then performing RNA sequencing. What she found from that experiment was that lincRNA COX-2 was the most upregulated lincRNA of all lincRNAs, as you can see on this outer track here in the circles plot. So this is what catalyzed an in-depth research to lincRNA COX-2, and thus my project. It was found by my grad student mentor, Alexa Robinson, that the highest expression of lincRNA COX-2 is in the lung and alveolar macrophages. So what she did is she did a qPCR experiment to quantify the expression of lincRNA COX-2 upon LPS stimulation within the lung. And as you can see in this qPCR bar graph, upon LPS stimulation, there's much higher expression levels of lincRNA COX-2. There are actually two types of alveolar macrophages, resident and recruited. Upon homeostasis, residential alveolar macrophages reside within the lung and have an anti-inflammatory response. Upon stimulation, recruited alveolar macrophages are signaled to be brought into the lung, hence the name recruited, and have a pro-inflammatory response. So what I'm looking at is the comparison of two alveolar macrophage cell types transcriptomes at days 3 following acute lung injury. So acute lung injury is the sudden onset of respiratory failure, pulmonary edema, and poor oxygenation. As you can see in this picture here, upon LPS stimulation, there's an increase in infiltrating immune cells and cytokines. Now here's a little depiction of what the project is that generated my data from my experiment, the RNA sequencing experiment, but I'll skim through that to save time. So the big question I'm trying to answer is if lincRNA COX-2 expression is specific to resident or recruited alveolar macrophages. So I was able to answer that question by utilizing a tool called RNA sequencing. RNA sequencing is a high throughput technology that provides unique insights to the transcriptome. RNA-seq can tell us which genes are turned on in a cell, what their level of expression is, and what times they are activated or shut off. So this is a general RNA sequencing pipeline, but once again, to save time, I'll be just skimming through it. The most important step, or the final step of RNA-seq, is differential expression. The goal of differential expression analysis is to highlight genes that have changed significantly in abundance across experimental conditions using the negative binomial distribution. So reading that becomes very apparent of why I chose to utilize RNA-seq to process this data. So what I found was that infiltrating immune alveolar macrophages express lincRNA COX-2 35 times higher than residential. This was very exciting. And here are my conclusions and future experiments. So as a whole, lincRNA COX-2 is highly expressed in alveolar macrophages. There are two populations of alveolar macrophages, resident and recruited. lincRNA COX-2 is more highly expressed within recruited, alveolar macrophages than it is in resident. So for future projects, I'll explore more kinetics of lincRNA COX-2 by assessing data from different days. Different days meaning 6, 9, or 12. So far, I've looked at day 3. All right, thank you.